Yes, good evening, all the participants. So, welcome to the day three session on biological databases. That's on data file formats, metabolic pathway, motif, and domain databases. So, before going to today's session, can we recap the last session contents? So, what exactly did we discuss in the last session? So, last session contents. So, I think some doubts are with Shilpa. If so, you can ask me. You can type the questions in the chat box. So let us start with a recap of the last session contents. So good evening to everyone. Yeah, the participants can interact so that even I can interact you more, or else I have to limit based on your understanding. Very good. So I got from first participant that we have discussed on how to use the UniProt database. A UniProt database is the largest database for protein information. Okay, so that is a fully annotated protein database, and this database is majorly governed by UK government. Also, Switzerland, Swiss government and Germany also is supporting this UniProt database. And I also got from another participant that we have discussed about genomes, especially on human genome. We discussed on the size, shape of the human genome. And then we have discussed on biological database. Very good. And we also discussed on biological sequences. Also, we discussed on genomes and proteomes. Okay. And related to pathway, we have gone to the KEGG. Can someone expand me what is KEGG? What is KEGG? So in this website, we can find good. We have discussed on protein-protein interaction. We discussed on NCBA. We discussed on UniProt. We have discussed on GenBank. Also, we came to the CHEMBL, and then we have discussed on OMAM, disease gene networks, the gene network. Very good. The expansion is Kyoto Encyclopedia of Genes and Genomes. Very good interaction from nearly 10 participants. How about the others? Can I go for the next question? Like, mention me any four databases present in cake. So, we have seen plenty of databases, nearly 8 to 9 databases we have seen in cake. So, tell me any four major databases you remember in cake. So, for protein sequences, we have gone for NCBH and PEPT, and we have gone for UniProt KB. Okay, so for protein sequences. And for nucleotide sequences, I have taken to the GenBank, NCBA GenBank. Okay, and if I take you to the cake, what are the major databases we can find? Four major databases. So we have discussed on, very good, on compound, genes, metabolic pathways, diseases, and what else you remember? Reaction, and then any other thing? Enzyme, reaction enzyme, and of course networks. Okay, so these are some of the important databases we are discussing KEGG. Good. So we have discussed all this, but what else is uncovered? We'll be discussing it on today. So if you see this, okay, on the second day session, we have discussed on sequence structure, genome specific, and special databases. Now we discuss about various database file formats, metabolic pathways, motif, and domain databases. Yeah. So before that, what is uncovered? So if you see this DNA sequences in NCBI, we have GenBank. So the source, major source, we take a pen. So this is from NCBI. And similarly, the GenPept is a database from NCBI. And if you see similarly, the SwissProt is something linked with UniProt. There is a major website called UniProt, isn't it? Where you have annotated and unannotated. I mean, you have, I show you directly from the website. If you go to the UniProt, .org. If you type for any specific protein like insulin, I zoom in. So I'm typing a specific protein called insulin, or we also have a keyword called insulin. Just clicking insulin, click search, as we're getting human insulin. If you see this, we have reviewed information and unreviewed information. The reviewed information from Swissprot, and this also have unreviewed information from Translated European Molecular Biology Laboratory. This is an exclusive database. We have EMBL for which is a repository of nucleotide database. If EMBL is a repository for nucleotide database, EMBL repository for protein sequence database. Okay, and if you see the reviewed information, you can find in Swissprot. So now I let it take you to the PPT. So I was discussing on various databases and what is the major source for this. Likewise, so I told you TREMBL and Swissprot are found in Uniprot, isn't it? This and this you can find in Uniprot. And before this PIR stands for Protein Information Resource. Protein Information Resource. So this is a major database. Okay, we find in Uniprot and few more major biological databases. And PSD stands for Protein Structure Determination. Protein Structure Determination. Okay, so this is a purely a protein sequence database. And of course, the NR, what we call it as non-redundant database. Non-redundant database, which we can find in NCBI BLAST-P protocol. If you try to if you try to use a software tool called BLAST, especially if you go to BLAST-P program, one can find this kind of a database. So even if you go to the few more classes, you can understand. You can able to see these kind of a databases. Okay, whatever covered, we can discuss. In protein structure, we have a largest database called PDB, isn't it? We also discussed on one database present in NCBA. Can someone tell me what is the 3D structure protein database present in NCBA? 3D protein structure database present in NCBI. What is the name? I showed you on yesterday like how to download a 3D structure of a protein. Very good. The answer is MMDB is perfect, not OMM. OMIM stands for Online Mendelian Inheritance in Man. So this is a database where you can find about genetic diseases and disorders. If you're working on genetic diseases and disorders, one can go to this OMM database. Again, if you see this OMM database is present in NCBI. Okay, so it's not a motif, it is an MMDB, Molecular Modeling Database, present in NCBI. Okay, so if you see this PowerPoint, okay, so I just mentioned PDB, but we also have MMDB. And gene expression, if you want to find gene expression data sets, especially the outputs derived from the microarrays, can be stored here. Even RT-PCR data, whatever gene expression, can also be stored here. So actually, whatever we get practically, okay, so all these data we can find in NCBI. So if you're if a medical from a physician, if he's prescribing some drugs and if the patients are taken the drug for treating particular disease, so the records, medical records of a patient are also stored in ClinVar. ClinVar, clinical variants. Okay, so one can also see that. So not only the biotechnologists can get benefited with NCB, even the physician, the doctors, MBBS doctors, whatever data they get as in the form of a records, patient records, that also is found in NCBA. Not only you find PubMed, the PubMed is meant for, you can find PubMed, majorly can find biotech literatures, biotechnology literatures, but also linked with pipeline. In medical, this is basically, you can find medical literatures, medical papers. So, research review, types of review, mini review, short review, all these papers you can find in NCBA. Okay, so NCBA is the largest repository. And if you see what are the databases linked with it, because if you know, if you go to the database, I take it to the Uniprot database, where the Uniprot is linked with so many databases. It is linking with PDB, it is linking with protein interaction database, it is linking with disease database, it's linking with the chemical compound databases, it's linking with the binding sites information databases, it's interacting. So you have much interacting much information can be annotated through the hyperlinks present in that database, isn't it? So if you see what are the major databases and what exactly or the hyperlinks you can find, how the nucleotide and protein sequence are associated. This is an example like how the bioinformatics databases are interconnected. If you see the DNA sequences, okay, this is from NCBA. In NCBA, we have GenBank, isn't it? And this is, of course, maintained by US government. And if you see this EMBL, EBA, this is maintained by Germany government. So you have an exclusive URL for EMBL, of course, even for EBA. Okay, and if you see this, you find the nucleotide sequences. How you find the nucleotide sequences in the form of GenBank and NCBA. Here you find generally a nucleotide sequences like GenBank you can find. And if you see this in NCBA GenBank, okay, you can also link with a protein database called GenPep. This is for protein sequences. And whatever protein sequences you're getting from labs, you have a right to even deposit in TREML, just like how you're going to deposit the sequences. If you want to deposit the protein sequences or nucleotide sequences in NCBA, one can use the deposit tools like Pancake, Sequin. There are two tools present in NCBA to deposit the sequences. Yeah, participants can mute. Yeah, so anyone can able to deposit any sequence. Okay, so let me continue that. I was discussing something on bioinformatics databases and connections. So I just discussed like in NCPA we have GenBank. Can have an hyperlink for a protein sequence through GenPep. And similarly for a nucleotide we have EMBL. We can have an interconnection with protein sequence called TREMBL. And even the GenPep can be linked with a protein structures information. If you want to get some detailed annotation of your protein, you won't get much information from GenPep, but one can able to go through PIR. So I already provided you the expansion, protein information resource, protein structure determination. So all these three databases, okay, are from NCBI. All these three are from NCBI. And if you see this EMBL, TREMBL, I told you this, all these databases are maintained by Germany government, also linked with Swissprot. Okay, that is, I told you the Switzerland also linked with uh, the Germany government to form Uniprot, isn't it? So all these things, okay, are linked with linked with EMBL or Uniprot, however it is. Okay, because even you can find the TREMBL everything in this Uniprot website. Okay. Now, let me take you to the next. Actually, this is actually a data taken in 2010, it seems. But now the size is rapidly expanded. You know, in GenBank, we have around billion sequences, billions of nucleotide sequences are deposited around. Okay, and GenPept is around millions. Okay, some 100 millions of sequences are deposited in GenPept. So the largest database you find is the GenBank, next GenPept, both are from NCBI, and then you can find TREMBL, Unigene, PRPS, this is brought always. Okay, so we have a large repository of sequences. So only we call bioinformatics data as the big data. You have plenty of genomes are deposited. Actually, I was explaining something about the human genome on yesterday. And when I'm explaining about human genome, I have taken you to the NCBA genome. Okay. Like NCBA genome, in NCBA genome, the genome is constructed from a person, only one person. But if you see this genome, okay, we have types of genomes. Like, for example, we can have pan genome. The pan genome is a collection. Okay, a pangenome is a collection of nearly 47 different racist humans. That is actually, if you see overall in the world, okay, so we can tell like Asian, Asian American, Native American, Black American. So we have types of people, isn't it? So based on the ethnicity, we can classify. If you see in the world, we can classify into majorly into 47 categories. Like how I told, okay, Asian American, and even can find Australian, and even in Australia, you can have Australian Indian. So we have 47 categories of human. So from each and every human, like for example, if you take an Asian, they have taken only one genome. And if you take Asian American, they have taken only one genome. Like this, they have accumulated all the 47 genomes and then they just mapped a single genome. From the 47 humans, they mapped a single genome. That's what we call it as a pan genome. So why they have mapped such a kind of a pan genome? Why they have constructed a pan genome? Because if one has to discover a vaccine, for example, I was mentioning you on SARS-CoV-2 vaccine, you remember? Yesterday, I was discussing about Covishield and Covaxin and the efficacy. The efficacy for the first strain of Covishield is around 82% after second dose. 82% after second dose, after administration of second dose. And if you see the Covaxin, it is around 91%. This is just for first strain of SARS-CoV-2. But later we have all beta, gamma, delta, so many strains came. Okay. So in that strains, if you see the specific delta strain, which is originated in India, the efficacy of the Covishield and Covaxin is less than 20%. Is less than 20%. See, drastically the efficacy is reduced in treatment. If so, then if you see even whatever the vaccines, Covishield and Covaxin, are majorly manufactured in India, even whatever it's going to target, the first strain of SARS-CoV-2, it targets only the Indian population. So whatever vaccines prepared here, if you try with the American population or Australian population, it won't give much efficacy. Got it? So based on the ethnicity, there were vaccines or drugs are developed, most of the drugs, gene targeting drugs, etc. So one should have an idea about the other genes, or about all the ethnicity, the genes of the different ethnicity people. So for this, they have constructed pan-genome. Not only that, okay, this is just a collection of 47 genomes, human genomes, isn't it? We have 1,000 genome project. We have 1,000 genome project. So if you click this 1,000 genomes, it is taking you to the corial.org. So in the 1,000 genomes, so if you just focus on a single genome, and if you're focusing on a rare disease, whether it will be getting a rare gene from the a single genome, it's very difficult. Or even if you are to get from 47 human genome, pan genome, even that is difficult. So one can increase the size. If you increase the size up to the 1,000 genomes, if you want to check with any specific disease like a cancer diseases, not rare diseases, you can try with cancer diseases. Okay, you may get some kind of precise results. Like you can come to know what is the exact gene responsible for causing the cancer for a particular patient. Okay, so this can be used for the treatment of diseases like cancer, whatever data is deposited from your 1,000 human genomes. Not only we have 1,000 genomes, this was rapidly expanded to 1 lakh genomes at present. Even we have 1 lakh genome project. This is established by Genomes England. So UK government has come up with a 1 lakh genome, in 2019, before pandemic. They come up with this kind of an initiative because they want to find even syndromes and rare diseases. They want to find what is the exact gene responsible. If so, if any patient has sequence, so they can blast with this 1 lakh genome, they can find what is the exact gene matching with it, and we can able to diagnose based on the gene targeting, etc. Okay. So in 2019, we have established up to 1 lakh genomes. One can search up to this. Now this is expanding rapidly. Even they are focusing on 10 lakhs, and it goes like that. So obviously, one can understand the database is obviously is going to expand, and you have find much big data only in bioinformatics. So if you take an hospital, okay, in a Chennai, a single hospital in Chennai, so in a day they can create around 10 genomes. Then how about for 365 days? Okay, if they're admitted for treatment of cancer, obviously they ask to come up with a genome. So you can find plenty of genomes are generated throughout the multi-specialty hospitals. So even there is a burden to have all these data in a server because the size that obviously matters. Okay, this is something about the databases, especially I was taking you about the genome. Okay, we have discussed about pan genome, we have discussed about 1,000 genomes, 1 lakh genomes, and even it's going to expand now. Not only we have 1 lakh genome for the human, we also have genomes established for rice, Oryza sativa. Okay, for few species we have, okay, which is most essential. But 1,000 genomes obviously you can find in the rats, mice, tomato, Solanaceae family. So we can find all this, okay. But 1 lakh genomes, very few, which is most important, like human rice, you can find. Okay, related to the databases, what else you can find databases in NCBI? I was explaining on yesterday also, that is, we have nucleotide like HN bank, and for a genome, we have an NCBI genome, we have a complete genomes. Okay, we have a taxonomy database, we have a structure database called molecular modeling database. Okay, domains, concept domain database, 3D domains, online Mendelian inheritance in man, single nucleotide polymorphism, sequence tag sites, gene expression omnibus, population study data sets, unigene, homologene, cancer chromosomes. So we also find NIH is linking with so many databases. One such is cancer chromosomes. Okay, so related to cancer, you can find a largest database even in NCBI. And gene expression pattern in mouse, protein, PubMed Central Books, etc. So these are something about what are the tools and databases present. So that about the tool databases you can find in this slide. And now we can go to this. And before going to the databases and data file formats, just an information to you that we are creating an opportunity to pursue PhD in Anna University under my guidance. I welcome PG, that is MSc, MPharm, ME, MTech aspirants or can able to register PhD in Anna University. So you can do research on in silico in vitro and in vivo studies. We have an excellent infrastructure on the effect of plant compounds on types of cancer, diabetes, etc. So the PG is, I mean, PhD scholars newly admitted can get around 18,000 to 20,000. Okay, as a stipend. And of course, I have two active funded projects, one from DST and AICT. So one can afford chemicals for your PhD research from this funding. Okay, now let me take you to the subject data file formats. So what are the different database file formats you can find in NCBA? In NCBA, we have Gen number for nucleotide, Gen info ID, PubMed ID, locus name. So what are that exactly? If you take the GenBank ID, I mean, you can have a locus name like for human beta globin, we can able to give like baby. So this is a keyword. One can you can also abbreviate like for insulin, you can give INS. So likewise, you can also abbreviate which you can consider as a locus name. And even the species, this is one such locus name. Beta globin is another locus name. So whatever give as a keywords or considered as locus names. Okay. Now go to the next slide. So in the next slide, we can find GenBank accession numbers. So the GenBank accession number is alphanumeric. And what is this point one? This point one denotes the version. Okay. Now even generally the accession number is 8 characters in length. But now that is in the recent 4 years, you can find even 10-digit numbers. So whatever recently they discovered, we can also find accession number with 10-digit alpha numeric letters. Now let us go to the next identifier called GenInfo ID. GenInfo ID is generally denoted by GI, and this is a 8-digit number. This is a 8-digit number given for a nucleotide or protein sequences. Nucleotide for for a linear DNA, circular DNA, as well as for mRNA, you have this kind of 8 letter, I mean 8 number GI, gene info ID. And we also have PubMed ID. So the PubMed ID can link a sequence, can also link the article in which they publish the sequence. Even they can publish an article. So for this, we can have PubMed ID. And what are the various database formats we can find? So if you see in NCBA, for a sequences, we have FASTA, GenBank. Yeah, even if it is a nucleotide or protein, we call that as GenBank format. But for a nucleotide, okay, we have a database called GenBank. For nucleotide, we have GenBank. Then how about for protein? For protein, we have GenPept. So this is the database, no doubt in that. But this is the format. If this is the sequence format, even GenBank is a sequence format for both nucleotide and protein. Likewise, we also have SwissProt format, ASN.1 format, and XML, Extensible Markup Language. And if you see the FASTA, how a FASTA format looks like, yesterday I told you that it's going to have after GA, whatever followed is a GenInfo ID, and after GV, whatever is followed is the GenBank accession number. So this is an alien number. So you got just 6 characters followed by the version. But now we are getting even 10 numbers. And this is a locus name. Okay. And this is also locus name. That the species name is also locus name. BRCA1, breast cancer antigen 1. That is the name of the protein. This is also a locus name. And of course, what is this? mRNA or protein. This is an mRNA sequence. This is also locus name. So these are the database identifiers. Generally, the database identifiers are present in first line. The remaining or the sequences or present in all these are things are should be in the second line. But the sequences are wrapped. The sequences are wrapped like from 1 to 60 characters will be present in first line. No, obviously 61 to 120 is present in second line. So it is wrapped for our convenience to read out the characters in the sequence. And GenBank. So the GenBank format looks like this. Okay. So I think I've shown you on the yesterday. SwissProt format looks like this. So it includes annotation. It includes the annotation information. So we can find much information even in Uniprot. And this is Genbank, Swissprot, and XML. The XML is actually extensible markup language. So actually, this is a database language where you can find user-defined tags. If you want to store the locus, you can use this kind of a user-defined tag. If you want to store the length, you can use this kind of a user-defined tags. So one can write the user-defined tags to store the data. So this is what a data, a locus name. This is the length of the sequence. So these are the data which is present inside the open tag and close tag. So this is open XML tag. This is a closed XML tag. Like how you find a HTML. HTML is meant for design language. For designing web pages, we are using that. But this is used for storing the data. Inside the open and close tags, you store the data. Okay. So this is something about XML. You can also find these formats for the sequences for storing the sequence information. Now, biological pathway databases. In biological pathway databases, first we have discussed on cake. So this is an example of pathway for hepatocellular carcinoma. So in hepatocellular carcinoma, so what are the major proteins responsible for causing the cancer? So it can find all this. Okay, and different pathways also it can find. Okay, how these pathways even that you can find. Here you can find RAS pathway, RAF pathway, and here you can find AKT pathway. So what are the proteins interacting with AKT pathway, PA3K pathway? So one can go to the cake.jp, and if you go to the pathways, and if you simply type hepatocellular carcinoma, you can get a kind of image like this. Then you can zoom in and you can see that information. Yeah, so related to string database. So yesterday I just taken you to the string. If not, I will be taking it to at present. So string database is for protein-protein interaction database. Okay. So string DB, you can give like string DB, it take you to the functional protein association networks. Inside that, go to search. Okay. So after going to the string-db.org, I'm going to click search and you're getting this kind of a window. Type a protein, what you're doing research at present. Now I'm typing a protein called tumor necrosis factor receptor associated protein 1. This is a kind of a heat shock protein 75. HSP 75 is also called as TRAP1. I'm going to search for this protein. So when I search for this protein called TRAP1, okay, I'm getting an information that whether you want to go for Homo sapiens. But we also got 261 more matches. But if your research is on Homo sapiens, that is, you're focusing on specifically TRAP1, which is HSP 75. Okay, that's what exact we got. So click continue. So if protein is found, you can find this information. If protein is not found, check for the related proteins. Okay, not all the proteins are deposited in string. But still, you can find 20 billion interactions are there and nearly some millions of proteins are deposited here, millions of protein interactions. Total, like if you see here in this protein, you can find a tryptophan is linked with 10 important proteins. The tryptophan is my hub protein, and these are the associated proteins. If you want to know what is pink 1 protein, simply click here. So you got some information about the pink one. It is actually serine threonine protein kinase. Okay. Likewise, if you want to know about a specific protein, one can click that kind of circular icon and you can get this. Now you are getting something like EXT1 is exostaucin 1. Okay. So this is something about how to find a protein-protein interaction. When there is a need to go for the protein-protein interaction, this most important is like when you are focusing on drug discovery for a particular target, if a trap an important target for a particular cancer, but if your drug, whatever you're going to give, can just inhibit the trap one, and if it is not going to bind with most closely related proteins, okay, which is suppose think like this HSP A5 is upregulating the trap one more, then even this is of course an important target with a hub. So whatever drug you're going to give, that should target TRAP1 and also it should target HSPA5. So not only that is based on upregulation, even downregulation matters. When compared to HSPA5, which is upregulating, you can find AHSA1 is more downregulating. Then compared to HSPA5, which is more less upregulating, one can take this AHSA negative regulating as the important target. So after TRAP1, you can also consider this as an important target. So based on the values, okay, based on the P values, upregulation, downregulation, one can decide that. Even the upregulation, downregulation can also be distinguished with the colors of the edges. Each and every protein, each and every protein connected in the network, we call this as nodes. We call this as nodes, and whatever lines, that is a distance between a protein, okay, from one protein to another protein, we call this as edges. So these proteins are interlinked through graph theory. So through the graph theory, they just got this kind of an information. So to know more about what wet lab studies are used to get this kind of an interactions, one can go to the viewers. If you click viewers, you can find the experiments, what experiments are made, conducted in wet lab to have these kind of interactions. When I go to the experiments, you can able to see the types of experiments they carried is anti-TAC co-immunoprecipitation. And we can also find anti-bait co-immunoprecipitation, pull down assay, so pull down assay. So likewise, we can find different assays are used. In each and every assay experiment, you can find the data also. According to anti-TAC co-immunoprecipitation, okay, you can find the TRAP1 is almost linking with 1557 plus nearly some 8 or 10 proteins. So you have 1567 proteins are interlinked with each other during the anti-TAC co-immunoprecipitation experiment. And in another experiment, that is anti-weight co-immunoprecipitation, you find 1047 plus 6 proteins. Nearly 1050 proteins or interactions are established here. So each and every experiment tells you how the proteins are interlinked. Okay. So this is some information on experiments, and you can go to the databases, networks, etc. But if you don't want, if you don't want to limit the interactions to 10 interactions, if you want to increase the interactions to top 50 interactions, to know about what are the top 50 interactions with the trap and can also increase the size of interactions, okay, by using this parameter called 50 interactions. If you want to increase up to the 100, click custom value and type 100. Now I want to give 15 interactors because if you give the size, you increase in size, it takes more time to load. Okay, so even if you have a good internet speed, you can find this information. I just going to give for 50 interactors and going to update. So I can find the top 50 interacting proteins with TRAP1. So this is our protein. TRAP1 is interacting with 50 important proteins. So if you want to download this information, one can go to the exports. So after going to the exports, if you want to download this, you can download in the form of the tab separated value. So this is actually an Excel file. Excel file, you know that you can download in the form of .xls, but here you don't have XLS. Instead, you have TSV, which can also be opened in Excel. Okay. So generally, what Excel you can use, you can download this TSV. If you click this download TSV, you can open it in Excel. And the same, instead of opening it in Excel, if I want to open in Network Visualizer, one can download Cytoscape. Cytoscape is the software tool. So if you get this kind of a network, you can't able to show this network in the publication because it leads to plagiarism. Because this data is already present in database, you can't take a snapshot and put in the publication. But the same, if you try to open it in a Cytoscape tool, Cytoscape visualizer, network visualizer, you can able to make it even some kind of a changes, whatever you want to make, like a coloring, some kind of visualization, whatever you make, you can make it, and you take that as a snapshot. You can take a snapshot and you can try to use that figure for publications. Got it? So don't take the data directly for publication, but you can use the softwares for publications. So this is something about the networks, protein-protein interaction networks, which is important for precision medicine design. If you want to get a precision medicine design for a cancer or any other diseases, one has to go with system biology approach. System biology approach, one of the major study is protein-protein interactions. Okay. So this is something about thing, and also there is something called analysis. If I take you to the analysis, you find different kind of parameters. This or the filters. One can use these filters to know about the important proteins connected to networks. For example, if I am focusing on hepatocellular carcinoma, okay, uh, and I'm majorly focusing on liver. Okay, then I have to select the liver, isn't it? So I'm checking the organ where I can find the organ. No, tissue expression. Okay, I'm going to click more here. So in tissue expression, I'm going to check liver. If I click liver, you can able to find red color. Okay, and if I'm focusing on particular pathway, for example, if I'm focusing on HSP90 CD737 pathway, so this is a second filter I'm going to select now. Click, you are getting violet color. One is red and the other is violet. For liver, and this is for a particular compartment. And even you can select any pathway. If you want a third filter, you can also select a specific pathway also. If you're focusing on apoptosis, click this, you find in green color. So three filters I have selected. Now let you take. I don't find any kind of only one green filter is there. Only one apoptosis. Whatever selected in red color, whatever genes, okay, or pertaining to the liver, and whatever are shown in violet. So this gene HSP90AB1 is linked with HSP90 CDC3 37 interacting complex. Okay, this is shown in red and blue. So liver and HSP90 CD737 interacting complexes. But whatever you find in green and red is like the protein is linked with liver and apoptosis. That is CYCS protein. This protein is having both apoptosis and it's happening in the liver. Likewise, you can also find the information of interacting proteins through the protein-protein interaction networks. Okay, so of course one can able to find the positive negative regulation using p-values. Okay, I also have few publications linked with it. So later I can also share it to you. Now let me continue about this database. Just now I completed about the string database. And now functional enrichments, whatever I selected, and I'm getting a different colors based on that. I can able to find the common protein. Okay, so all these things are functional enrichments in the string database. So whatever I done, I just representing in the form of slides. One can also come to know what are the important genes responsible for a particular cancer. Okay, by selecting the various enrichments, advanced filters. So there is also another database called Stitch, which is a compound protein interaction database. Earlier, we have protein-protein interaction, but according to the Stitch, you can also find a ligand. This is a drug. Bifenthrin is a plant compound drug that is interacting with various proteins. XBB1 is a protein, MAPK8 is a protein. These are also proteins. So one can find compound protein interactions through Stitch database. Now, there is also a database called FunCo for protein-protein interactions. Okay. So if you want to take you to the Stitch and FunCo, I can take it fast. So first I'm going for Stitch DB. So the same thing I'm going to try. So can we try something different? No. Presently the website is not functioning, but we can go for Funko. If it is not functioning today, maybe tomorrow or day after tomorrow it functions. You can try later. But the just go in Google default type stitch, and then you can it take you to the first URL. Okay, now I've just gone to the Funko. This is a protein interaction database, but you can find a kind of a good interactions and you find advanced filters. The same if I'm trying with the same trap one. So I'm going to give a trap on protein. I'm clicking search the Funko. It takes you to the protein interactions of the specific protein what I type. If you want to try with insulin, type insulin and just check with it. So now you're going to get a kind of a network. It takes some time. So then I can just show you. So this is how we get an output for, okay, for a trapper. You can also use the filters like liver filter. You can also filter by pathway and you can get only the selected enrichments alone you can find in blue colors. Okay. And we have some databases like Sequin and Bankit for deposit of nucleotide sequences, especially if you see this Sequin, this is an offline software. That is, you can install Sequin in your operating system and you can, from the standalone software, you can upload the sequence. So what kind of a sequences can be uploaded? So if you see this, you can submit the complex submissions also. That is complex submissions of long sequences. But whereas a Bankit, if you see, this is an online submission tool. This is not an offline, but this is an online, a web-based tool. And generally, simple sequences can be submitted, not the complex submissions. So the difference between Sequin and Bankit. The tools for deposit of sequences. So this is a secondary database called CAT. What is CAT? CAT is actually class, architecture, topology, homology. So the expansion CATH. Okay. So the proteins motif, the motif is nothing but a part of a protein which gives a function or a structure of a protein. Okay. So what is the most important part of a protein which gives the structure or function? So that whatever you have motifs, you can categorize, you can group it in the form of secondary databases. One such database is a CAT. Okay, like CAT, you have another database called SCOP. This SCOP is structure classification of proteins. In Google, if you simply type for the CAT or SCAP, the first URL can direct you to this kind of a databases. So one can able to see the proteins are classified based on 7 category of classification. In CAT, you have 4 classes. I mean 4 categories of classification, but here you can find 7 categories of classification. Okay, these are also of course secondary databases. Genome specific, you have plenty of databases. We have plenty of databases. Even in cake, we have a genome database. Like if you want to know about the genome, okay, you have uh, for solanation. So this is how a Funcook database looks like. Jiggling. Yeah. So just I'm going to select something like a liver. I'm going to select a pathway called PA3K pathway. So wonder whatever is selected at present. Okay. What happened? The system is slow actually, but whatever is selected alone or related to this pathway. So okay, then I can take you to the next database. What I was discussing on genome databases. So you have separate databases for the family also. You know, you heard about the Solanaceae, that is tomato family. The tomato, ashwagandham, everything belongs to this Solanaceae family. So for a Solanaceae, you have a separate genome. For Drosophila melanogaster, you have a separate genome URL. So for Sol Genomics Network, not only you have a for a family, you have a separate network. You also have a separate genome for the tomato. Also, you got it. You have, these are some of the databases where you can find for family of proteins, a family of plants, and also you can find for a single plant also. For even for a tomato, we have a separate website for a genome. Got it. So if you're working on plant research, one can go to this specific URLs for the genomes. And what is this NHRI, National Human Genome Research Institute? So one can find much information of the human genomes in this institute. That is, this is maintained by NIH, National Institute of Health. The URL is genome.go. NIPGR, that is National Institute of Plant Genome Research. So one can go to this URL to fetch the information of the genome. And if you want to collect any specific gene targets for a cancer, for a specific type of cancer, one can go to this mycancergenome.org. So these are other public domain databases. Always remember NCBI, UniProt, PDBJ. These are, of course, largest databases. You find plenty of information. But if you are specifically focusing on a disease like cancer, asthma, you have specific databases. If you go with a cancer genome, there is a largest database called mycancergenome.org. This is not linked with NCB, but this is a database for cancer genomes. Likewise, if you want to go specifically for a cancer, a specific type of a cancer, for hepatocellular carcinoma, I have a TBMHCC. For this, you have this kind of a database. So you can find such a kind of plenty of databases even for a specific disease. So not only for the cancers, for even autoimmune diseases, for infectious diseases, we can find plenty of databases developed. So this is a database developed from India. A famous scientist called Raghava has developed so many tools and databases. Even myself, I am working in protein bioinformatics lab under the mentorship of Michael Krumiha. So he is India's number one bioinformatician. If you want to know about him, okay, like IITM Protein Bioinformatics Lab. So if you type also PBA Lab IIT Madras. Okay, so if you have a knowledge on development of tools or development of databases using machine learning techniques or some bioinformatics approaches, so one can get experience in this lab, protein bioinformatics lab, to know more about what projects are completed, what kind of tools and databases are developed. Okay, one can click here in the resources, you can find databases and tools. Okay, so if I click databases, see, there are databases like a curated database for binding affinity and utilization profiles. These are the new databases, like in the present era and previous decade. Okay, in the previous decade and this decade, these are the databases established. So one can get flourished while working on these kind of related researches. Okay, so this is specific kind of a database. Like if you see MPAD, a database for binding affinity of membrane protein complexes and the mutants. RSIM, it's a RNA small molecule interaction miner. In his lab, he has worked on development of databases for RNA interactions, RNA protein interactions, carbohydrate protein interactions. So many interactions he come up with different databases. So there's a famous database called ProThermDB, a thermodynamic database for proteins and mutants. Okay, so you can find plenty of databases inside. Not only databases, each and every database is a product of a PhD scholar. And similarly, tools. So like Raghava, scientist, okay, even Professor Michael Komi has developed so many tools and databases for the interactions, especially through the machine learning techniques. So you can find that kind of a tools. So if you want to work on this kind of tools, okay, you can go to this website and you can work with it. Okay, so I was explaining you like on about few scientists who has developed the tools and databases. to work with. So not specifically to the cancer, but also to the other diseases. These databases are in common. These can be used for common applications. And this is, of course, the largest database for cancer genome, C-Bio portal. If one has to find detailed information of the RT-PCR analysis of the cancer patients, and for multiple patients, like nearly 100 patients, 1,000 patients, want to find information, or if you want to find information for a specific hospital, like John Hopkins Hospital or a center. Okay, so if you want to find about the samples information of a particular disease, so here I search for liver hepatocellular carcinoma, I'm getting information on that. Okay, so if you track down, you can find much information for the cbioportal.org. OMIM, this is of course also linked with the NCBI. You have an OMIM database, but you can also go to the exclusive URL, omim.org. So that here you can find information of the genes related to genetic diseases and genetic disorders. Okay, so you can find the chromosome locations. If you click that, you can direct to the chromosomes also. The genes mapping to what chromosomes, that information you can find. Of course, the national NIH is also taking care of maintaining a National Cancer Institute GDC portal, that is Genome Data Commons Data Portal. So the URL for this is portal.gdc.c. In each and every URL we are linked with NIH, you don't find NCBI dot. You have an exclusive thing, but these are also maintained by NIH USA, National Institute of Health USA. So if you want to work on specific kind of a cancer, it can be a lung cancer, which is prevalent among males and females. So one can go to the specific, find the cases, and you can find the genes. Even you can find the specific location. Okay, in your specific location, what is the major gene affecting the lung cancer? All this information I can find in GDC portal. Gene expression omnibus from NCB to know more about the microarray data. Gene cards, of course, this is a very famous database for genes. So, like if you want to more information of the protein, I suggest you to go with UniProt, isn't it? For detailed protein annotation, one can go with UniProt. Am I audible, participants? Yes?
Yes, sir.
Yes, yes,
Yes,
okay.
sir.
Sorry, because I'm not asking
Yes,
you any
sir.
question. I want to check whether I'm in streaming or not. Okay, good. Thank you for your interaction. Yeah, gene codes. This is the largest database for the genes. Okay, so when compared to the NCPA, you get confused with the NCPA if you want to check with the genes. If you want to know what are the genes responsible for hepatocellular carcinoma, just like that, I typed that as a keyword and searched, I got nearly 8,100 results. This is the best website for any diseases. If you type a disease name, and you can find plenty of information on that. You can click plus and you can find full detailed annotation of the specific gene. Much information you can find if you expand each and every gene. So all the genes, full annotation you can find in gene cards. The cancer proteome atlas. For the cancer proteome, one can go with tcpaportal.org. Okay. And you know that even in uniprot.org, you have a proteome database. We have a proteome database even in Uniprot. And related to targets, like if you want to focus on specific genes, okay, one can go with open targets, GPA. Okay, these are some of the databases. And this is a database I developed in SVC, Sri Venkateswara College of Engineering. That is an adjuvant or immunoanalytical database. Actually, PG scholars in 2017, they have developed this database from C#, which is used in the foreground, and SQL, which is a database, and PHP personal homepage for web page applications. Okay, so we just worked on developed in .NET framework. And what exactly is this database? Generally, we have deposited the docking data, molecular docking data. Where we got the molecular docking data? We got docking data from Discovery Studio. There is a software called Discovery Studio. We had in our institution, we have a commercial software. So we use this database. I mean, we use the tool for docking with the docking plant-based adjuvants with dendritic cell receptors, surface receptors of dendritic cells. And we got around 1,000 interactions. All these 1,000 interactions, we just updated in a database. So we use these options like update, we add the data, all these things we used. And later, if you want to search a particular thing, like if focusing on autoimmune disease and how to combat autoimmune disease, then if you type a specific search parameter, you'll be getting what is the best plant-based adjuvant we can use for the treatment. Even in the COVID, the SARS-CoV-2 vaccines, the plant-based adjuvant, the best plant-based adjuvant used for vaccine is saffronin. Saffronin is the best adjuvant. Generally, in adjuvant, we go for alum. But other than alum, if you want to reduce the accumulation of metals, because that is a metal oxide, isn't it, alum? Instead of accumulating metal oxide in our body, nowadays they are trying with plant-based adjuvants. Okay, so now based on that research, plant-based adjuvants, we have developed a database, and uh, so this is something about today's chapter, that is on data file formats, metabolic pathways, motif, and domain databases. If you have any doubts, you can ask me. Do you have any doubts? MSc subject, uh, MSc subject, no, like actually, I don't understand. Yes, yes, sir. Okay, do you have any doubts? If you have any doubts, you can ask me. Unmute and answer. Yes, for projects, yes, I am guiding MSc students, MTech students, for PhD also, and even for MSc also, I am guiding projects in the form of consultancy. In 2021 or 22, from Sri Krishna Arts and Science College, Coimbatore, two students came here and worked projects under my guidance. And immediately after completing their projects, on the next month, they got job in IT company, okay, related to the projects. So for projects, sir, MSc with subject, any related to biology, anyone can do the projects. UG internship, yes, I'm also guiding projects for the UG. Now, of course, if you know in the PBA lab, I am one of the current member. I'm a postdoctoral research fellow. So I not only linked with IITM, also having an incubation center in Ramachandra Medical College. Okay, so I'm also having incubation center in Ramachandra Medical College, and also linked with Satyabhama Research Park, and linked with NIRT Chennai for the project. So I linked with multiple institutions. Okay, so there won't be any problem for access of infrastructure. Of course, we do have a best infrastructure. We have an approved animal house facility, um, and so both animal, we can work on animal experiments. We can also work on cell culture experiments. Yes, MSc Zoology is eligible. So one PhD scholar with this qualification is working under my guidance. Yeah, everyone, MSc biology related, everyone, MSc botany, zoology, everyone are eligible. Yeah. So any other doubts you have related to the thing? So if not, so on the tomorrow, we'll be discussing on sequence analysis. So what is sequence alignment, types of sequence alignment, and about amino acid substitution scoring matrices, we'll be discussing it on tomorrow. Okay. Thank you then. Okay. Thank you for your patience and interaction. So tomorrow we can try to interact as much as possible because a lot to discuss. So it is better.